Let's start. Good morning, everyone. Thank you for joining us for today's webinar. My name is Patricia Aguilar. I am the chair of the ACAV subgroup, and I'm co-chairing this webinar along with a member of our trainee leadership group, including Rachel Lange, Megan Boat, Livia Saqueto, Amy Christosic, Maria Oñango, and Isabella Mauricio Resende. We're very thankful to have the support of the National Basketball Association for this webinar. They have partnered with us today with the goal of sharing and disseminating correct information about SARS-CoV-2 and variants. So before we introduce our speakers, just a little housekeeping. If you have any questions during the presentations, please type them into the Q&A box. And we will bring them up for discussion during the Q&A, and we will have time for questions at the end as well. This webinar will be recorded, so if you miss anything, don't worry, we'll be posting this webinar on the YouTube channel. And now I will ask Rachel to introduce our first speaker.
Good morning, and thank you everyone for joining us. My name is Rachel Lang, and I'm a doctoral student at the New York State Department of Health. And I'd like to introduce our first speaker, Dr. Mauricio Nogueira. Dr. Nogueira received his MD and PhD from the Universidade Federal de Minas Gerais in Brazil. And he then was a visiting fellow at the NIH in Bethesda for 4 years before returning to Brazil, where he's currently a professor of infectious diseases at the Faculdade de Medicina de São José de Rio Preto, and is currently the president of the Brazilian Society for Virology. So whenever you're ready, Mauricio, feel free to share your screen and start.
Hi, Rachel, thanks a lot. Just a small correction, I'm not the president of Brazilian Society for Virology anymore. I'm not now, but I used to be. So I would like to thank the invitation from the ACAV, and I'm really glad to be here, actually here in my home, but I would love to be with you guys at some meeting. We are all missing seeing each other and discussing each other. I'm discussing science with each other. I'm pretty sure that you are seeing my screen right now. So I'm gonna talk a little bit about what SARS-CoV-2 did in Brazil. I'm sorry, trying to see, okay. So this is the beginning of this nightmare that we are living right now. This was the first manuscript that describes this virus, SARS-CoV-2, and the beginning of the outbreak in China. This manuscript, together with a second one that was published a few days later, describing the clinical aspects of the disease, they start to take our attention for something that in a couple of months become the biggest public health issue that we saw in this generation. So a lot of effort was diverted from other researchers and from other groups to SARS-CoV. And of course, my lab is arbovirus lab, but since the beginning, we move some effort to try to respond this outbreak in several different aspects. In Brazil, we have a few programs that were working with emergent virus. One of them was CADI. We'll talk a little bit later about CADI, but this CADI program, that is a Brazilian UK program for emergent virus, and I'm a part of this, we quickly move our perspective to see what was going on in Brazil. So we start to look this. First, we look for the disease, COVID. And the first case in Brazil was detected on February 2020, on 2050. And after that, we analyzed the first 3 months of the epidemic in Brazil. And we saw that the number of cases of COVID-19 starts to increase here on the epidemiological week 12. That's pretty much February. But even before that, there was a huge outbreak of acute respiratory disease with unknown etiology. And we know for sure we're talking about SARS-CoV here in the COVID. But from where this virus got in Brazil, and of course, the introductions in Brazil in the important cases during the first weeks of the outbreak were exactly from where Brazil has more commercial and ties, especially US was responsible for 22.5% of all introductions in Brazil. Of course, detected introductions. Italy was the second one, Spain, UK, and Portugal. And of course, we had a government in Brazil at that time. This is the same that we have right now, that don't think about science, but thinking about politics and saying, okay, we should avoid travel to China, but China was not responsible at all for the important cases in Brazil. Also, the virus got in Brazil really fast, and even in the number of cases increased even faster because we have a basic reproduction number in Brazil that was higher than Europe at the time. So we could estimate that it's about three in Brazil. But even inside Brazil, there was differences in some states like Amazonas, Rio, and São Paulo when you compare with Ceará. And why Ceará have a different, a lower reproduction number is still under debate, but we can say for sure, and I'm gonna show you a little bit later, that they have a different strain of virus than the other parts of the country. And we also soon in Brazil try to implement the non-pharmacological interventions. And in fact, they were quite successful in changing the trends of the transmission. You can see here in Amazonas, we see an exponential curve that tends to be a little bit lower. And Ceará, as I said, it was completely different, the situation from the other states like Rio de Janeiro and São Paulo. After we look for the disease, we also looked for the virus. This is another manuscript that was published by this program, the CAD program that we are part of. And in this work, we look for the virus, not only for the disease. And Brazil was one of the fast growing epidemics in the world. And this can be easy to explain due to large size, large population, and some level of sophisticated economic that relies a lot on travel inside the country. So after the introduction of the virus, and as I show you, when we look for the disease, we had that reproductive number about 3. When we sequence the virus and estimate, we have pretty much the same number. And again, as you can see in São Paulo, the basic reproduction number goes quite steady around 3. And when we start the no pharmacological interventions, this reproductive number drops at some points below 1. But unfortunately, these no pharmacological interventions, after a couple of months, the people decide that they don't want this anymore. And we unfortunately were not able to really implement this for a long term. So these numbers start to grow again. When we look in the beginning of the virus circulation in Brazil, we see a pattern that something until March 1st, most of the cases were imported cases. And then we start to have a second wave of transmission now with virus that were circulating inside the country. And what is important that there was several introductions, but at the beginning, only 3 clades were responsible for most of the cases in Brazil. And clade 1 that was the most prolific clade was pretty much a clade that was in São Paulo state. The clade 2 was widespread in Brazil, and the clade 3 was a clade that was only present in Ceará state. So this might explain the differences between the basic reproduction number and the some difference that we have in Ceará regarding pathogenesis, at least we had last year. So this is a snapshot of the beginning of the outbreak in Brazil in the last year. It should be important, and it's quite obvious that at the first epidemic phase, most of the cases were in the big cities in the central south Brazil. But the second epidemic phase last year, most of the cases were dispersed from these big centers in Brazil, mostly São Paulo, Belo Horizonte, Rio de Janeiro, and Ceará, then here in the north. And these cases were, these cities amplified the virus and disseminated through Brazil. I will apologize, I will have 3 slides in Portuguese, but just to show how this is affect our lives and our institutions. This is a bulletin from my institution. We are issuing this bulletin daily since March last year. So this is from Saturday. So as Saturday, we have 209 patients in my hospital, and at that point, 121 patients in intensive care only in my hospital. And this brought us a lot of load in the lab. So my lab, both in the hospital and the medical school, so last Friday, we analyzed 960 samples by PCR. And as you can see, a little bit over 25% positive. But if you take from the March the 50th was the first day that we did a PCR here for COVID, to 26, we did 182,000 PCRs for COVID. I probably, my lab, in other labs that I had in my life that I worked, we haven't done this amount of PCR for everything else combined. So that was a lot of pressure that put in our labs. In my hospital, we have admitted so far 3,700 patients, and we unfortunately lost 820 people died in my hospital. And of course, since we are a lab devoted to dengue and we move everything to work with SARS-CoV and we live in an endemic area, we also see sometimes the overlap of these diseases, like we published this, a fatal stroke doing a dengue and SARS-CoV co-infection in a patient here. He first went to the hospital with dengue. He was discharged with 2 days of the disease, and 2 days later he came again, now with SARS-CoV, and had a stroke. So we have now a Brazilian network of COVID genomics. This network is being shared now by Fernando Spilk, that was my successor at the Brazilian Society for Virology. It's funded by the government, federal government, and this program has, I'm sorry, yeah, some problems here. We are trying to collecting samples all around the world. We have some genomic centers that are doing sequencing and submitting this to databases, and we are trying to study all aspects that can be evaluated from these samples. So we are now 12 institutions in Brazil, my institution, and in others like Fivale, Fiocruz, and Amazon, in Belo Horizonte, in Brasilia, several campuses of Unesp, Unicamp, USP, and other ones, and the National Center for Scientific Computation that does the bioinformatic works for us. So pretty much we generate data, we submit to this, the clinical data also to this national lab in bioinformatics that held all this. They do variant analysis and they issue reports. So this is, all these reports are public available. So anyone of you now can go to this site, Coronaomica BR, NSCC, and have a clear view on what is going on in Brazil regarding the variants of COVID here. So we have graphic of the most frequent mutations per geographic region. And of course, this has been leading us to find some variants of interest. This is one of them that was published by the Ana Vasconcelos and Amilcar Tanuri groups, showing this new lineage of SARS-CoV in Rio de Janeiro. And other lineage of interest that of concern is this one that is being described in the Amazon region with mutations, the E484K and N501Y in the spike protein. This lineage has been subject to a lot of controversy in Brazil because of a supposed higher transmission rate. And I would say supposed because no assay was done regarding the viral fitness of this strain. So we are just doing some inferences based on epidemiology, and I should take this with a grain of salt. One of the other programs that we have is the CADI group program. That's the Center for Arbovirus Discovery, Diagnosis, Genomics, and Epidemiology. So this group was funded by FAPESP, that's São Paulo State Agency, and by MRC in England to investigate arbovirus emergence. But of course, you put a lot of effort in SARS-CoV. So we were looking for epidemiology, genomics, and how this affects the environment and transmission of this disease. So this is an example, the work done by, I showed you before, 2 manuscripts that we published at the beginning of the outbreak, and this is being done right now. Was just released by Nuno Faria and our friends from CADI, showing that this new variant in Manaus has a higher attack rate than expected. As you can see, there was pretty much what you can call a clade replacement in November last year, November, January. So you can see most of the cases were from other lineages. This lineage emerged at some point in November, December, and they pretty much are responsible for all of this. So this lineage emerged in November and now is being dominant in Manaus and is now being circulating most of the Brazil. And something that we also could, Nuno and his group figured it out, and something that is also indirect reference for better viral fitness, maybe a better viral fitness, is that the CT values in the patients with this lineage are lower and significantly lower when compared with other lineages. So this needs to be addressed soon. So as I said to you, this Coronaomic network is giving us real-time reports, and we have these 2 variants of concern circulating in Brazil. P1, that's despite the fact that it's most restricted to Amazon, was detected in the other places, but this second variant of concern, the P2, with this E484K mutation is being widespread, has a widespread circulation in Brazil. So we are in a continual effort for sequence surveillance of this surveillance of the emergence and the spread of the variants. But we really need to do in vitro studies because we want to see how these viruses are neutralized by convalescent sera, and most important, by individuals vaccinated. We already vaccinated a few million people here in Brazil, not as fast as I would like, but we are vaccinating. But we are now also seeing, and we saw last week, the first time that someone vaccinated got in ICU care. And we are sequencing the virus and try to see if it's a different virus or was just something from this special patient. We also need to do in vivo studies with these strains, take a look with more care for these patients. And of course, we need to do, we want to generate clones for that, and maybe Pei-Yong can help us a little bit later. So I would like to thank Nuno and Fernando Spilke, Nuno from CADI, and Fernando Spilke for our coordinator of the Corona Omics network, for sharing some original data that I'm sharing to you. And I'll thank the Brazilian government by the Ministry of Science and Technology, CNPq, and CAPES for funding, as also FAPESP and NIH that funding my lab on a regular basis. So thank you very much.
Thank you, Dr. Nogueira. Super interesting talk. Just a reminder, if you have any questions, just submit them in the Q&A chat on the side and we'll address them at the end. So our next speaker that we have is Dr. Nathan Grubaugh, and Dr. Grubaugh is an assistant professor of epidemiology at the Yale School of Public Health. He spent 7 years in the biotechnology industry monitoring food production lines for pathogens and also developing early phase vaccine candidates. He then earned his MS in biotechnology from Johns Hopkins University, and also then went on to earn his PhD in microbiology from Colorado State University in 2016. He then did a postdoc at the Scripps Research Institute to study the 2015-2017 Zika virus epidemic, and his research currently uses genomics, to determine the emergence risk and to track the spread of mosquito-borne viruses like Zika, chikungunya, dengue, West Nile, and more recently, SARS-CoV-2. So specifically, his lab sequences viruses during outbreaks for epidemiological investigations, determines disease phenotypes and transmission fitness of novel viral mutations that occur during outbreaks, and maps the evolutionary pathways that a virus takes to adapt to new environments. His research goals are to integrate genomic data into surveillance and response programs to better prevent and control mosquito-borne and zoonotic viral outbreaks. So whenever you're ready, Dr. Grubaugh, please feel free to start.
Thank you so much for a terrific introduction. Thank you to the organizers for inviting me to come talk with you. When I submitted my title a few weeks ago, just because of the very rapidly evolving topic, didn't actually really know what I was really going to be talking about. But I think my title does a fairly decent job now. What I'm going to do is just sort of take you through the lens of some of the data that we've been seeing and some of the work we've been doing that is primarily to help build tools and provide recommendations for surveillance and for public health actions with the SARS-CoV-2 variant specifically in mind. To be a little bit more specific here, so most of my talk is going to focus on what is going on in the United States, with a very heavy focus then on the variant B.1.1.7 that was first detected in the United Kingdom. I'll touch on a little bit of B.1.351 that was first detected in South Africa. And then, as Mauricio just gave a fantastic introduction for, with P1 that was first identified in Brazil and also Japan. But to focus here on B.1.1.7 for a second, what is high interest of this variant is this cluster of all these different amino acid substitutions that are in the spike protein. And I think we all know that the spike protein is very, very important for entry into host cells and for eliciting an immune response. And of a couple of the mutations that are of high importance here is one, this 501Y mutation, which I know you can't see in there because everything is sort of packed together. And also now some of these B117 viruses in the United Kingdom have independently evolved the E484K mutation that Mauricio also mentioned, that is found in the P1 variant and also B1351. Of course, now there are also lots of other variants of interest that aren't quite yet defined as variants of concern. Those include P2 that Mauricio introduced, which P2 is also found throughout the United States and it has the E484K mutation, but we don't quite yet know its true public health impact. You also may have heard of the California variant, which is B1427 and 429, and also B1526 that was recently described out of New York. But again, I'm going to really focus on B117 because I think that is the most pressing need, at least from the US side of things. B117 was first detected in the UK. Interestingly, it was the researchers in the UK were alerted about B117 really because of the program that was conducted in South Africa, where they discovered this mutation 501Y in the spike protein that seemed to be associated with a lineage that was spreading faster. And then therefore the researchers in the UK started looking for it in their data, and they found this lineage B117 that had all of these different mutations in it. And then as they started plotting this and tracking this variant, they saw that it was rapidly taking hold and becoming the dominant virus in the UK. Some of those early studies looked at both the sequencing data, the frequency of B117, but also data from a PCR assay that I'll bring up a couple of times. It's called the TACPath assay that serendipitously helps to detect B117 because one of the three targets, the one specifically in the spike gene has a target failure because there's a deletion that sits in a probe site. So that doesn't detect the variant. It's still positive for the test because the other two targets are positive, but it's been used to help track. But anyways, some of the model estimates then suggested that B117 is 50% more transmissible. Also, it leads to a 0.4 to 0.7 higher reproductive rate. In this really great study done by Public Health England, where they were looking at individuals that either had B117 or didn't have B117 and determining if they were transmitting to close contacts, they found that B117 had a 30 to 50% higher secondary attack rate. And this figure that I'm showing here of the map is from early January, so it's very out of date, just a month and a half or so later. But since its first detection in the UK, B117 has been detected in, I believe, 94 countries, including the United States. The first B117 case in the United States was reported, I forget the exact date, but it was right before New Year's Eve. It was from the Colorado Department of Health. And then a day or so later, it was also detected out of California from Christian Anderson's group. And as of February 22nd, now we have over 1,600 reported B117 cases. And of course, that number is probably much, much higher than that because the only way to actually determine if it is B117 is if you sequence the virus. But most states that are doing sufficient or some sequencing for variants have detected. So you can essentially assume that B117 is in all 48 of the continental US. And what is concerning is that B117 may work to reduce this trend that we've been seeing of decreasing cases. So of course, in the United States, we had this huge spike following the holidays and at the beginning of the new year that has been going down rapidly. What I'm concerned about is this leveling off or perhaps even an increase in cases. And This is, we can't attribute it directly to B117 at the moment, but that is one of the worries that this might help reduce that trend. So hence why we are so concerned and wanting to be vigilant about trying to control this variant. And that's because we know that it's on the rise in the United States. This is a figure pulled from a colleague's paper where they were looking at rates of tests that were determined to be B117. And while this was for this data goes back to a month from now, so it's a little bit old, they projected that based on this data that B117 was 35 to 45% more transmissible, and that its frequency or proportion was doubling about every 10 days. And so if we were to look at today, it would suggest that the proportion of B117 cases in the United States would be somewhere around 20%. And while I don't have the data in front of me to show you, I've been seeing this. So last week, I saw a lot of data, and I'll show you on the next slide that B117 in many locations was around 10%. I've seen some new data that is very recent that would suggest that maybe it's between 15 to 18%. So I think that this trend line is holding up. And that is important because if it continues on this rate of doubling every 10 days or so, the estimate is within about a few weeks that B117 will be crossing this threshold of being the dominant virus in the United States. So I'm at Yale, and Yale is in Connecticut. So I do most of my work here in Connecticut. My group works very closely with the Connecticut Department of Health and other local health agencies. Here is a map of Connecticut. So New Haven's right in the middle here of New Haven County. These are the B117 cases that have been confirmed in Connecticut through sequencing. Most of these are found in New Haven because that's where we do most of our work. And just over the last 3 weeks, and again, I don't have last week's data up here yet, I'll probably have that soon, you could see the proportion of cases that are B117 has been going up significantly, and it certainly fits that maybe 7 to 10 day doubling rate, where last week we already, at the end of last week, we already saw that it was at 10%. And I imagine that that will be somewhere in the 15 to 20% range at the end of the reporting period, what would have been Saturday. And because to confirm these cases, you have to do that by sequencing, we end up with a lot of great sequence data that we can use for epidemiological investigations. And for this, honestly, it doesn't make a whole lot of sense to just work in a vacuum within your own state. It made a lot of sense for us to collaborate with other groups so we can incorporate other data and to have a much broader picture of what is happening throughout the US. So we work very closely with the CDC, with the New York Department of Public Health, the Wadsworth Center, the University of Michigan, and then many, many other colleagues in Yale and also from around Connecticut. The Connecticut genomes that we had sequenced are these large purplish pink circles here. And that is combined then with these sequences from many other US states. My lab also does a lot of sequencing from Illinois and New Jersey that are shown on here. But the main point that I wanted to show with this slide, as the title suggests, is that there's been many separate introductions of B117 in the United States that have led to sustained transmission that is evidenced by the clustering of these sequences. These branches that are grayed out and in the background, these are genomes from the UK or other parts with it from the world. So you can see again that we have many different introductions that have occurred. Working with our collaborators again over at Scripps and Helix from and mainly California and Florida data. This figure on the left here shows the densities around when introductions of certain clades likely happen. So I'm not showing the phylogenetic tree here, I'm just showing the estimates of when they think that there's a large clade from California and from Florida, another one from California, when they are actually introduced into the United States and started transmission. And they estimate that B117 was successfully introduced between late November and early December. when we looked at our data here, also we have Florida data, and we also see that this is a late November, early December introduction. When we looked at some data from Michigan, we see that this is early December, also New York, New Jersey, again, early December. So we have multiple evidences now of introductions that led to sustained transmission that happened at least a month before it was first detected. Then also, that is very important, by late December, we're finding evidence for interstate domestic spread. So we have this clade here that was introduced into New York, and we find transmission that has led to a cluster of cases in Connecticut, clusters in Michigan as well. And also from Florida, we see evidence for transmission that happened to Illinois. I have many other examples of this, not showing on this slide here. But again, all of this happened before the first B117 case in the United States was detected. So again, that first detection was somewhere right before New Year's, and we had now evidence for multiple introductions leading to community transmission throughout the US and interstate transmission of B117 in the US before we actually had real evidence that was in the US. And I think that's a really big problem for our public health agencies when they're essentially blind to what is happening. And so the flashlight for them is surveillance. And what we need are more flashlights. And so this graph here shows the number of COVID-19 cases in the United States that have been sequenced and shared on GISAID. And this is from the CDC, they update this monthly. So I imagine either today or tomorrow we'll have a new update that goes all the way out to March here. And before calling attention to where we need to do more work, I would also just really like to applaud the efforts of all the groups that have been sequencing. I think it's quite impressive that within a year that there's been 120,000 SARS-CoV-2 genomes that had been sequenced. It's really, if I were to rewind a year and think about that, I mean, that is sort of an unfathomable number. And it's really amazing work by a lot of these groups that are doing it. Of course, the CDC and public health labs have been putting a lot of effort to this, but also a lot of academic labs whose job it is to do surveillance. In academic labs, we are used to testing hypotheses and doing things that are specific around around projects, really, to get papers, because that's what we need for PhD students to graduate, what postdocs need to move on and get additional jobs, and what we need for to get grants. And so it's really hard to put a lot of effort directly into something that is really just a public health service, and that is surveillance. And so again, I just really want to applaud all the groups that have been doing all of this work. It's expensive and tiring. But again, as I said, we do need to do more of it. And of course, across the US, I think we need to increase surveillance, but there's also some places where we need to do maybe a little bit more. So again, this feels like outdated analysis now because it's from a few weeks ago, but to determine where does surveillance need to be increased the most, and this is specifically now for B117, we started by looking at between December and January, this important period for B117 introductions and the start of community transmission, what percentage of the cases were actually sequenced? And we saw that in this period, actually in the United States, only 0.13% of the cases were sequenced. Why is that number so much lower than what we've reported for the overall pandemic? And that's because we also had the highest number of cases during that period. And it's really hard for labs just to ramp up proportional to the amount of cases that are happening. We usually have a limit of what we can sequence. And so the overall percent is lower. Plus also sometimes it just takes a little while for data to make it onto repositories like GISAID. So I imagine this number will actually be a little bit higher. And if we look at, so the bars here show the percent of cases that had been sequenced. So you see there's a lot of heterogeneity among the states. Some states have done very little sequencing during this period relative to the number of cases. And some places like Wyoming and Maine are getting actually quite close to that 5% threshold that we need. I think they're helped by the fact that they don't have as many cases as let's say like New York and California, for example, but still, I think this is really amazing effort. And then these black dots represent the number of B117 cases that had been sequenced during this time period. But to know where specifically surveillance would have to be increased, at least for B117, we also then have to think about where do we expect B117 to be transmitting to most, or where would we expect the most number of introductions that would have happened during this early phase. Caveat here, this figure is used using airline data from October. We now have airline data from December to update this with, but we started by just looking at passengers that are inbound from the United Kingdom, because that's where the larger proportion of B117 cases are in the world. But obviously there are other places now that also have B117. And then we just map the number of passengers arriving at each of these airports. And you can see some clear areas where we're getting the most number of passengers around this New York, New Jersey area, Chicago, California, Florida, and also Texas. So then we looked at the numbers of passengers arriving, and we compared that to the number of sequences on GISAID. And thankfully, the places where we have the most number of passengers arriving, so perhaps the highest likelihood for B117 introductions, are the places where we sequence the most, and that's New York and California. And now if we look at this same sort of plot, but look at just the number of B117 cases that were sequenced, that we see that states like Texas, Illinois, and New Jersey have a relative few number of B117 cases relative to the amount of travel. And if we look up here, there's also not as much sequencing that is happening. So this suggests, but doesn't necessarily prove, that if you were to increase sequencing in these locations, that you would see a disproportionate number of B117 cases that were underreported in states like these, and probably other places too, even though of course B117 is underreported across the board. One thing that you should see that stands out as sort of interesting is that the number of cases in Florida that were sequenced is actually quite low relative to the number of B117 samples that have been sequenced. And that's because there was primarily done using a targeted approach. And I mentioned earlier this TAC path assay that has an S gene target failure with B117 that helps to identify cases. And this, and while we need an unbiased approach for sequencing for B117, this has been great for helping to identify Florida as an early hotspot. And it helps us also to think about, are there non-sequencing approaches that can aid variant surveillance? Sequencing is the obvious gold standard here, but it's expensive. Not every lab can do it, and sometimes it's not scalable. So we need to think about other approaches. I'm highlighting the SARS-CoV-2 genome and a couple of really key deletions. And this first one here is this deletion called 6970 in the spike protein. This is 6 nucleotides that are not, that are deleted in B117 and other viruses. If we look at this phylogenetic tree, all the sequences that have this deletion are colored either yellow or orange. And when we look at the blowups of these, we see that here exists B117, but also is this other lineage B1375 and B1258 that aren't really thought to be of public health importance. So while you're detecting B117, it is unable, the TAC path assay that just has this as a dropout is unable to differentiate between these 3 viruses. So we went and looked at all of these. And so also importantly, it is unable to pick up P1 and B1351. So if you're only using this S gene target failure as a screening assay, you will not detect these other variants. So we went back and compared the genomics of all these variants and found that there's another deletion one in ORF1A that is a 9 nucleotide deletion or 3 amino acid deletion. And these are all colored by the sequences that are blue and orange. So we see that P1 has this deletion, B1351 has this deletion, and so does B117. But these other two that aren't of concern that gets picked up by just this doesn't have it. So it's actually able to differentiate. So we created an assay that we've now shared on protocols.io. We have a preprint on it that we're working on updating it right now. And it uses, I'm just going to go back for a second. It uses a target and the nucleic capsid as a control. And it uses these two targets. And the idea is that if it's not a variant of interest, that you will detect all 3 of these targets. And if it's one of these, you will find dropouts in either of these while this one is positive. So I encourage you to try this out if you're interested in it. We have some supplies that we can mail out to labs that are in LMICs to get you started. And that's exactly what we've been doing lately, is that we've been starting to ship these reagents out to different labs. Importantly, we've been shipping these to our friends in Brazil and South Africa to validate the assay for P1 and B1351. And here's some data. This shows these other lineages, so things that aren't necessarily of concern. And when they don't have either of these ORF1 or spike deletions, that we have detection of all 3 targets. When the sample is B117, we find detection of N1, but we don't find detection of this ORF1A or spike. With B1351, we find dropout of ORF1A, and also with P1, we find a dropout of this. This guy here turns out it was a mislabeled sample, and that has been corrected. And I was told as of this morning, we have a much higher N here. And we plan on updating our preprint that is currently on medRxiv with this new data any day now. And I put this slide up here just as a remind myself to tell you all that we wrote this commentary. I wrote it with some of my friends and colleagues with expertise in a lot of different realms of public health on what are some suggested public health actions to use to control SARS-CoV-2 variants. I'm not going to go into detail all of those things. I encourage you to read it if you want more information. But one of the things that came out of that, besides obviously needing more surveillance that I just talked about, was how we end up controlling a variant like B117. To control that, we really need to be able to answer this question of how is it more transmissible? And there's a few different options here, and they're not mutually exclusive. There could be some combination of these 3 things that make it more transmissible. But for simplicity here, if B117 is more transmissible, either because it has a higher viral load, meaning that when people are infected, they produce more virus, or if because it has a decreased infectious dose, meaning that you don't need as many infectious viruses to infect you. With both of these situations, if these are true, for public health actions, this is where things like double masking or increased ventilation in indoor places or additional closure of indoor places is going to be needed to control the transmission. If B117 is more transmissible, not because it has a higher viral load or decreased infectious dose, but just because you're infected for longer, the control measure might be to increase the period of isolation. We started investigating this through our partnership with the National Basketball Association, who are also sponsors of this symposium today. But they have one of the most unique cohorts in the world, in that the individuals associated with the NBA are getting tested daily. So from each infection, we have the first positive case all the way up to the peak, and then all the way through clearance. And when we combine all this together, we can actually, and we work with Jonathan Grod's group at Harvard, we can look at these proliferation periods, these clearance periods, the peak infection, the duration of infection. And we started by comparing symptomatic and asymptomatic individuals. But as B117 is increasing throughout the country. There had been some cases within the National Basketball Association, which allowed us to then look a little bit deeper into the dynamics of B117. I'm just going to start and maybe say this a couple of times. Huge caveat here is N equals 7. Obviously we need a lot more data to really investigate this. So first is just looking at the peak viral load. And you're probably used to seeing this where RNA copies are on the Y, it's on the X this time. So if it's to the right, it means there's more virus. The dots show the individual samples. So red are B117 cases and blue are non-B117. And we see that most of this overlaps. So maybe B117 with these has a little bit higher viral loads, but because they overlap so much, it's hard to conclude anything from that. What stands out a lot more is the duration of acute infection is longer with these B117 cases than with a non-B117. If we put that all together, again, caveat N equals 7, is that we find the duration of infection for non-B117 cases is 7 to 10 days. And what I mean by duration of infection here is days PCR positive. So that may not be duration of infectiousness. But for B117, we find this is quite a bit longer. This is between 10 and 17 days. And so the peak infection is not really much different, but this proliferation stage and the clearance stage takes longer to occur. I think that is good on time for this. I'll be happy to answer some more questions at the end. I thank you all for coming to this talk and listening. Here are some of my acknowledgements. It is laughably small in terms of the total number of collaborators that have actually worked with us on this. I bolded some of the members of my group that have worked tirelessly for over a year, trying to do all that we can to help our community in New Haven and to create tools and products to help throughout the country and throughout the world. Also, our many different collaborators. I'm just naming some of the groups of the heads of different groups that we've worked with recently on some of this work that I'm sharing here. Definitely want to give another shout out to the National Basketball Association and the Players Association for being really great partners in research and funding a lot of our work. And thank you.
Great. Well, thank you, Dr. Grubaugh, for that very interesting talk. And just a reminder, if you have any questions, you can put those in the Q&A feature in the webinar, and we'll deal with those at the end of all of the speakers. Quick introduction, I am Megan Vogt, and I'm a postdoctoral researcher at Virginia Tech, and I'm very excited to introduce our next speaker, Pei-Yong Shi. Dr. Pei-Yong Shi is a professor in the Department of Biochemistry and Molecular Biology and John Seeley Distinguished Chair in Innovations in Molecular Biology at the University of Texas Medical Branch. He's also an elected fellow of the American Academy of Microbiology, adjunct professor of emerging infectious diseases at the Duke NUS Graduate Medical School in Singapore, and honorary professor at the Wuhan Institute of Virology, Chinese Academy of Sciences. He works on RNA viruses, drug discovery, and vaccine research, and his unique experience in public health laboratories, such as the New York State Department of Health, pharmaceutical companies such as Novartis and Bristol Myers Squibb, and academia at UTMB and Yale, allows him to work on both basic and translational research. He has published greater than 300 peer-reviewed papers, and his group developed the first reverse genetic systems for the epidemic West Nile virus and Zika virus, and discovered flavivirus N7 and 2O methyltransferase activities. His team also published the first peer-reviewed infectious clone and reporter virus for SARS-CoV-2. Besides academic excellence, he also has a stellar track record of senior leadership role at major pharmaceutical companies, such as executive director at Novartis Institute for Tropical Diseases, where he set up antiviral strategies and executed drug discovery and development. He contributed to the development of fostemsavir, an FDA-approved HIV drug, and many of his technologies have been licensed to leading pharmaceutical companies for countermeasure development. A recent example of this is his reporter neutralization assay that has enabled the rapid development of Pfizer's COVID-19 vaccine, which was the first COVID-19 vaccine with 95% efficacy in humans. So with that, I'll turn it over to Dr. Shi, who will give us his talk entitled SARS-CoV-2 Biology and Countermeasure Development.
Thank you, Megan, and it's a great pleasure to be able to present some of our recent work. And well, first of all, I really want to thank the organizers for the opportunity to present. And what I'm going to do today is to talk about the reverse genetic systems, the older version versus the newer version. And it's really exciting that the newer version will allow us to perform the culture of the viruses at the BSL-2. And then also I'm going to talk about some of our recent work on the new variants. So as we know, the SARS-CoV-2 is a coronavirus that is a very long single-stranded RNA genome of positive polarity. It's about 30 kilobases in length. So when we initially started the reverse genetic system, we pretty much copied the previous technology by RT-PCR7 fragment to cover the whole genome, and then in vitro ligate them to a whole cDNA about 30 kilobase in the test tube. And then on the very 5' end of the sequence, we engineer T7 promoters so that we can perform in vitro transcription to make the 30 kilobase RNA. And then once you transfect these RNA into cell, in this case, Vero E6 cells, we are able to recover high level of recombinant viruses. This is a plaque assay from the recombinant virus compared with its parental virus, which is the Washington state isolates. Soon after we developed this, we thought for application purposes, we would like to engineer a reporter genes into the viruses. Again, based on the previous coronavirus wisdom, we engineer reporters into the ORF open reading frame 7 of the virus. And as indicated by the red star here, in this case, I'm just showing you the amnion green, a fluorescent reporter was engineered at the ORF7, and we can recover virus as it's shown here. And then when you infect the cells with this reporter virus, you will see the cell turn to green. And then you can measure the intensities of the infected cells. And then you will be really able to track the viral replication levels using those reporter signals. So one of the first things we developed this for application is to trying to develop into a high throughput neutralizing assay. So the assay with the reporter virus is very simple. On day 0, you seed the cells into the 96 or 384 well format. And then the next day, you mix the anti-serum, or if you are interested in drug discovery, you can add the compounds and mix the anti-serum with the virus for 1 hour and let them neutralize, bind. And then you load the mixture into the pre-seeded plates and wait for 16 hours. After 16 hours infection, and then you just use the high content imaging to measure the number of green cells. And then this way you will be able to look at the inhibition. For example, this is without serum, and this is with positive serum containing the neutralizing antibodies. You can see the reduction of the positive cells, and you can plot these out in a very nice way, and you get the titration of the neutralizing antibodies from your specimen. So this assay, we very rapidly engaged Pfizer to support all their preclinical and phase 1, phase 2 neutralizing activities. And these are the packages for the FDA approval, EUA of the vaccine. So then the bottom line is, this initial platform was very authentic, but with its shortcoming that it's still the BSL-3 agent, and you have limited access to for many labs around the world. So then the question I've always asked for is there a way that you can make the culture and do it in the BSL-2? So recently we succeeded to achieve this goal as indicated here, and that we used a transcomplementing system which is being used for many, many viruses. So the idea is the following. So we delete 2 genes from the viral genome, in this case, open reading frame 3 and the envelope. And then we engineered, in this case, reporter amnion grain into the OFF7, as I just described in the previous slides. So then you have 2 deletions of the genes in the viral RNA. And then you make a transcomplementing cell that is inducibly express the 2 missing proteins, ORF3 and the envelope protein. So when you have these RNA into these transcomplementing cells, and they will be able to package infectious viral particles. And then, but the particles, the genome itself remains 2 genes missing, right? So when you infect the next round of a naive viral cells, for example, and they will infect the cells in an authentic way, just like wild type virus infection. And because the genome misses 2 viral essential proteins, and then they will replicate, but they can no longer form the next round of particles. So that forms a single round of infection, and that conceptually will allow us to bring it down to the BSL2. So this is the genome in panel B, shows how we deleted those regions. And then we also added some safety guard into the system. For example, the transcriptional regulatory sequences indicated in the green box were mutated to red, so that when this RNA, our recombinant RNA, if it's accidentally recombined with wild type viruses, happen to be in the lab or the people got infected, and then the resulting virus will be a lethal virus, because the virus to replicate, it requires all the TRS region to be identical in order to able to replicate. So now this is the first experiment we did by basically electroporating this RNA into these transcomplementing cells. And then you collect the supernatant, and you can see that the viral particles, infectious viral particles, are made. And then under the EM, you can see the particles. And then when you use these supernatant, the viral particles to infect the naive viral cells, and for the first round, you can see the cell lights up green, 28, 48 hours. But if you take supernatant, a culture fruit from these cells to infect the next round, naive viral E6 cells, you no longer see infectious signals, indicating that the particles produced in panel A is only a single round. But of course, there is, you can see the data, the limitation of this initial system is the titer is not that great, 10 to the 4th TCID 50 per ml. In order to improve the robustness of this system, what we did is we let these particles to continue infect, passage them on the transcomplementing cells in this cycle for 10 rounds, hoping that the viral genome started to adapt so that this efficiency of this transcomplementation will be improved. And this is the experiment we did. And you can see the brown bar is the 24, 48 hours of the initial particles, and the titer is 10 to the 4th, 10 to the 5th. And then when you passage them for 10 rounds, those are the green triangles, you can see it significantly boosts up the titers of the system. And if you sequence the genome of the green particles, and you will see there are 3 mutations accumulated in the genome. And when you engineer all those 3 mutations into the original virus, you can see now this is the black diamond. It can match the 10-round passage of the particles. Through this way, we're able to increase the efficiency of the packaging to high titers of the infectious, single-round infectious viruses. And then the red dots is just using the authentic viruses with the amnion grain. So it can really match very equivalent titers of the production. So then the next thing is about the safety or the virulence of these particles. So we use the two animal models to look at the safety of these single round particles. The first one is using the hamster model. We used intranasal inoculation. For all the single round infectious experiment for animals, we dosed the highest possible dose to look at the potential virulence of the particles. So here, panel B shows you the weight loss, and the triangle black is indicating the wild type virus, and that is 100,000 TCID50 inoculum. And then the single round particles, 6 times more infection dose, and didn't show any weight loss. It's just equivalent to the mark. And then when we look at the diseases of the infected animals, and the wild type infected animals show diseases. In contrast, the single round particles, there is no disease at all, the animals. And then when you look at the viral load in the nasal wash, oral swab, and the trachea and the lung, the single round particle RNA level is a couple of logs lower compared with the wild type infected animals. So then we, through collaboration with Mike Diamond, and we tested the system into the K18 human transgenic mice, and we did 2 routes of infections. The first route of infection is the intranasal infection. As you can see that the wild type infected mice significantly lost weight, while the single round particle infected animals it didn't lose weight at all. Again, look at the doses. It's a couple of hundred more dosing levels to start with. And then this is the lethality also reflects the situation. The wild type virus infected animals die, while much higher dosing inoculum of the single round of particles, there is no death. And then to increase the stringency of the testing, we performed the intracranial injection. As you can see, direct intracranial injection of the wild type virus caused the death, significant weight loss, and the death. Even at the 5 TCID50, it still caused some of the mice died. In contrast with a single round infection, intracranial injection, and there is no weight loss, no death. So based on this, the UTMB IBC has conditionally approved that this system will be able to downgrade it to be performed at the BSL-2. And then to test the utility of the system, we performed a few rounds of experiments. First, we tested 20 convalescent patient serum of COVID patient, and we tested them by 2 different assays. One is the conventional plaque reduction neutralizing assay. And then for each sample, the other testing is just using the single round amnion reporter signal to do the neutralizing assay. So these are the raw data. In panel B, it shows the correlation of the 2 assays. They correlate quite well. So that indicates this assay can be used for high throughput neutralizing test. And we also tested the monoclonal antibodies, just as example. In panel D, it shows very nice dose response. And then antiviral drug discovery, we tested the remdesivir in 2 different cell types, A549 express human S2, as well as the viral cells. You can see very nice dose response. So we're very excited that this system can be implemented to both academic and particularly pharmaceutical and companies. They will be very, very useful so that they can really do those assays at the BSL-2 setting. So now I'm going to move to the second part to talk about some virulent studies. So this is relevant. We're doing a similar experiment with the new variants right now, but as example, I'm just going to use the D614G mutation to show the system and how we work it out. So as we know, when the virus was initially sequenced, the SARS-CoV-2, the first 2 months at the D614 of the spike gene, it's it's all aspartic acid. As time goes on, you can see this position of the amino acid started to switch to glycine. And this is called the D614G mutation. So then the question is really biologically, does this mutation increase the viral fitnesses, like the previous two speakers both commented on that? And then also, does it increase the transmission? And the third question is equally important, does that affect the vaccine efficacy? So we used the reverse genetic system as well as the hamster model to interrogate these questions. So we engineered a pair of isogenic viruses, these are the mutations, and then the recombinant virus in terms of plaque morphologies, they're almost identical. When we infect the CALO3, which is lung epithelial cells, to do the growth curve, compare with the two viruses. The black is the original D614. The red is the mutated G614. You can see there are some level of advantages in terms of replication. And then if you analyze the same specimen over during the growth curve to quantify their viral RNA, you can see the mutated G614 is even a slightly lower. Then if you calculate the RNA PFU ratio, which is the parameter to indicate the infectivity of the viruses, it clearly shows that the mutated viruses, the variant sequences, is more infectious because it has a lower RNA per PFU ratio. So then we moved to look at, move the system into the hamster model to look at the pathogenesis and the potentially transmission of these two isogenic viruses. So we intranasally infect the hamsters, and when we look at the weight loss, both groups infected by original and mutated virus, they all showed similar weight loss. And when we look at the viral load in the different tissues of the respiratory tract, nasal wash, trachea, and the different lobes of the lung, you can see initially on day 2 post-infection, the new variant has a slightly higher viral load in the sample. And on day 4, it is clear that the nasal wash has significantly higher viral load in the nasal wash, while in contrast, the lung lobes, there is no difference between the two viruses. So that really indicates that this D614 substitution increases the fitness of the viruses, particularly, at least in hamster model, that increases the upper respiratory replication levels, and therefore potentially might be transmission. And then we also tested these isogenic viruses using the human airway culture, right? On the upper airway, you can, the upper of the transwell, it's really exposed to air, and only at the basal of the culture, it has the medium to maintain the cells. So panel B just shows the growth kinetics of the pair of viruses. Again, you can see the variant, these G614 is much higher compared with the Y-type sequences. And again, we look at the RNA levels using the same sample, and there is similar, they are similar RNA levels. So if you do the RNA PFU ratio, again, you can see the new sequence, the G614 is much more infectious from the samples produced. And then we did the competition experiment in panel E. So we mixed 1-to-1 ratio of the two viruses, inoculant, to infect these cultures. And then over the time, over the next 5 days, we take the produced viruses and analyze the ratio between the two viruses. You can see over the time, the G614 virus can efficiently outcompete more than 10-fold compared with the original virus. Even if you do 3-to-1 original input, and in this case, in panel G is 9-to-1. And then over the time of the infection, you can clearly see the G614 virus will win the competition in these human airway culture. So these experiments clearly demonstrate that the D614G mutation enhances the viral replication in the human airway culture. And at the same time, we also look at the stabilities of these viruses. So basically, we incubate equal amount of infectious viruses at different temperatures, 42, 37, and 33, which mimics the upper airway, our airway temperature. And you can see over the time, the red virus, which is the mutant virus, is much more stable compared with the original virus. So these data demonstrate that in terms of the thermal stability, the mutated virus is much more stable compared with the original virus. And that might contribute to the enhanced level in the upper airway. And then after our publication, two independent groups, one from Yushi Kawaka and then Ralph Barrick's group, they did using the hamster and performed the direct transmission experiment as indicated here. Clearly, the D614 mutant virus transmits much more efficiently, particularly at the early time during the transmission on day 2. And the similar results were reported by Dave Wentworth, Volker Thier, and Martin Pierce group. And they used both hamster and ferrets to demonstrate that the mutation significantly enhances the transmission between the animals. So then I'm going to move to the neutralizing activity. So then we tested the effect of these mutation on the neutralizing activity for the monoclonal antibodies. Here we tested in panel D a panel of monoclonal antibodies with defined epitopes on the receptor binding domain. And you can see really epitope dependent. This monoclonal antibody 18, it drops the neutralizing activity against the variant. And then through collaboration with Pfizer, we tested the serum collected from animals or human and that had been immunized with the approved vaccine. So mouse, macaques, and human, and these are the immunization schedule. And then you can see for both mouse, macaques, and human, they all have some modest reduction in terms of neutralizing activity. So these results demonstrate that the D614G enhances the viral replication in both animals and the human lung epithelial cells, and they improve the viral fitness in the upper respiratory tract and leading to enhanced transmission. And the virus, the mutation enhances the viral infectivity and the stability, and then it modestly affects the neutralizing susceptibility. And mechanistically, structural studies show that this mutation will promote the receptor binding domain to an upper conformation, which is the conformation required to engage the human S2 receptor to attach the cells and infect the cells. So now the last two slides, what I'm going to do is to show some of our very recent work on the African spike variants and their effect on the vaccine elicited neutralization and activity. So again, using the reverse genetic system, we engineer the full spike sequence with these mutations indicated into the Washington isolate. So the plaques are slightly smaller on the far right compared with the original Washington state viruses. And we also made some other mutants. In the interest of the time, I'm not going to go into the details. So then what we did is we used 20 patient serum who had been vaccinated by the Pfizer's vaccine. And then for each of those serum, we test them for their neutralizing activities against the original virus. In this case, we can just look at the panel A, the panel C, This is the black indicates against the original virus, and the red indicates against the virus containing the full spike of South African sequences. So then you can see there is a reduction. It's about 2/3 of the reduction once you have the full spikes into the virus. And I think I will stop here for, for the final acknowledgement. I need to acknowledge my lab and then tremendous collaborations with Pfizer and scientists over there. And then for the transcomplementation studies, we collaborated with Mike Diamond's group for the mouse experiment and amazing colleagues at UTMB and the funding agencies. And I will stop here. Thank you.
Thank you, Dr. Shi, for the amazing talk. So hi, everyone. Just a brief introduction of myself. My name is Livia. I'm a postdoc at Dr. Mauricio Nogueira's lab in the creatinine project, working with emerging arbovirus and virus host interaction, and also with SARS-CoV-2 genomics. So now I'm going to introduce our last panelist, Dr. Anna Durbin. Dr. Durbin received her Bachelor of Science in Pharmacy from the University of Michigan and her MD from Wayne State University. She is certified in the specialties of internal medicine and infectious disease. Following her fellowship in infectious disease, Dr. Durbin became a medical officer at the National Institutes of Health, where she spent 5 years using recombinant DNA technology to develop novel live attenuated respiratory virus vaccines. In 1999, she joined the faculty of the Johns Hopkins Bloomberg School of Public Health with a joint appointment in the School of Medicine. She has worked with NIH in the development and evaluation of live attenuated dengue vaccines and has served as the principal investigator for nearly 30 clinical trials evaluating these candidates. She has, in collaboration with NIH scientists, developed a controlled dengue human infection model to down-select candidate dengue vaccines prior to the initiation of large-scale efficacy trials in endemic areas. Dr. Durbin is the site principal investigator for the phase 3 AstraZeneca COVID-19 vaccine clinical trial and an investigator of the Pfizer phase 3 COVID-19 vaccine clinical trial. She has served on FDA and WHO vaccine advisory committees. She received a National Institutes of Health Merit Award in 2005, a National Institutes of Health Directors Award in 2011, and the Instituto Butantan Medal from Brazil in 2013. She has published more than 90 peer-reviewed papers. So now she will share with us her talk entitled The Impact of SARS-CoV-2 Variants on Vaccine Efficacy. What do we know? Welcome, Dr. Durbin, and feel free to share your screen and start.
Great, thank you very much. It's really my pleasure to be here and to follow the wonderful speakers that we've had so far. So I'm going to review a little bit about the vaccines that are currently in clinical trial and that have received authorization in several different countries around the world. There are differences in how these vaccines, the platforms that these vaccines use, and I'll review the efficacy data that we have. I'll also look at efficacy specifically against the variant vaccines. So this is a slide that just goes through the different vaccine candidates and platforms that we have. I think it's important to note that the mRNA vaccines and the virus vectored vaccines induce not only very good neutralizing antibody responses, but also because of the way they utilize the individual cellular machinery to present the antigen. They also have excellent induction of CD4 and CD8 T cell responses, something that subunit protein vaccines or the inactivated vaccines don't really do. So I put this slide up here, just so that you can see the spike protein and where the N501Y mutation is that's the mutation that's been found in these different variants. The B117 variant that was very well described in previous talks that emerged in Britain. And then we also have the 501Y period V2, which emerged in South Africa in December. We know that this vaccine may reduce the effectiveness of some vaccines, and I'll go through this variant. And then the P1 or the 501Y.V3, which emerged in Brazil in late 2020. We have all of these variants. Well, we've had reports of these variants in the United States, and I think it's important to note that these variants arise stochastically. So they're, because of the effects on viral fitness that Pei-Yong went through very nicely, they're able to outcompete the other strains or the other variants or other virus strains, and that they have arisen independently in many different places. So these strains or these variants don't need to be imported necessarily into different countries. They can arise spontaneously and have arisen spontaneously in many different parts of the world. So I'm going to go through the demographics of some of these vaccines because I think it's important to understand where efficacy studies of these vaccines were done and what the efficacy results are. So I'm going to start with the Pfizer vaccine because that was the first vaccine to receive EUA approval here in the United States and has also been approved in many other countries around the world. So this was a clinical trial that was conducted in the United States, South America, and South Africa, but nearly 77% of the volunteers were from the US. Only 6% were from Brazil, and I point that out when we go through the efficacy. Also important to note that almost 83% of participants were white. We only had 9.3% who were African American or Black, and 28% who were Hispanic or Latino or Latinx. If we go through the efficacy of the Pfizer vaccine, what we see is excellent efficacy. So overall efficacy of 90% against COVID of any severity. So that means any symptoms, you could have a runny nose and a sore throat and a COVID positive test, and you are a case. We also note very excellent efficacy against severe disease. We note very good efficacy here against COVID in the elderly, which is also very important, knowing that the vast majority of deaths, at least here in the United States, were in those people 65 and older. I do point out here that we had lower efficacy in Brazil. We don't have all of the strains sequenced, so we don't know if that's lower efficacy was due to variants or not. I should also point out that this efficacy is against viruses that really do contain the D614G mutation because that variant overtook the United States very early in the pandemic. And these efficacy cases were being recorded really from about September onward, when we know that the 614G variant was the dominant strain in the United States. If we now go to look at the demographics of the Moderna vaccine, it's important to note that this vaccine study was conducted in the United States only. 79% of the volunteers were white, only 10.2% of the volunteers were Black or African American, and only 20.5% of the volunteers were Hispanic or Latinx. So it's a very homogeneous population, again, only conducted here in the United States. When we look at efficacy, again, we have excellent efficacy for both of these vaccines. Again, I want to point out that this is demonstrating efficacy against the virus with the D614G mutant. When we look at efficacy in the elderly, again, it's excellent. It's a little bit lower at 86%, but really, really good efficacy and 100% efficacious against hospitalization, severe disease, death in the populations here in the US. Again, very good efficacy here against communities of color. And again, that's very important, at least when we're looking at the outbreak here in the United States, where the vast majority of the burden of COVID was borne by communities of color. And Pei-Yong went through this a little bit, but I want to go through it again just to highlight some of the neutralizing antibody studies that were done against the UK variant and the South African variant. Now, as Pei-Yong said, this was done using recombinant viruses where they introduced specific mutations into the USA WASH1 background. If you look at the different colors here, we have a mutant that represents the UK variant, which is here, and you can see very similar antibody titers to the UK variant with the Pfizer vaccine. And then down here, you have an E484K N501 D614G, which does represent the South African variant, but it doesn't include all of its mutations. And this was a paper published in Nature Medicine. So we see reduction in neutralizing antibody titers to at least the South African variant. They went on, as Pei-Yong showed you, to include and introduce more mutations. And so the B1.3151 spike contains all the mutations found in the South African variant. And what you can see here is a further reduction overall in the neutralizing ability induced by these vaccines, or induced by the Pfizer vaccine. So if you look at the wild type virus here, you see a neutralizing antibody titer of about 1 to 500. That is reduced about 5-fold here when we test the neutralizing antibody capability against the full South African variant spike. But what I want to point out is these are still very good titers. So you still have a geometric mean titer greater than 100. So it's still able to neutralize the South African variant, but not as well as the wild type or the UK variant virus. Now, the Moderna vaccine, they also tried to measure neutralization against the variant spike proteins, and they used a little bit of a different approach. They used a vesicular stomatitis virus pseudovirus neutralization assay, where they used DNA technology to introduce the spike protein into VSV and have that virus express the spike protein, and then see how well the virus could be neutralized. And this was a nice assay to do because you didn't have to do it in BSL-3. It could be done in a BSL-2 facility. And what you're seeing here is the B117 variant here, the D614 mutation alone. And they're looking in A and B match samples with B117, and then down here in C and D, you're looking at the full-length spike protein. So what you see is very little or very modest effect on the UK variant, and you're seeing a more marked effect here with the full-length spike protein of the South African variant. So again, neutralization of the B117 or UK variant was not affected to any significant degree. However, the neutralization of the B1351 or the South African variant was reduced, but the geometric mean titers were still greater than 100. So reduced, but still able to neutralize. And we know, at least from the rollout of the Pfizer vaccine in the UK and in Israel, that there is very good efficacy of this vaccine against the UK variant. We don't have real world efficacy data of either the Moderna vaccine or the Pfizer vaccine against the South African variant. We can only use these in vitro assays, but we do know that at least the Pfizer vaccine is highly efficacious against the UK variant, and we would expect the Moderna vaccine to also be highly efficacious against the UK variant. I'm going to go on to the Johnson & Johnson vaccine, which just received emergency use authorization here in the United States. And what I like about this trial and the data that are coming out about this vaccine is I think we can extrapolate some of the data, some of the information that we've learned about the Johnson & Johnson vaccine, and extrapolate that to the mRNA vaccines and perhaps to other vaccines coming down the pike. So this is a single dose vaccine, which I think is important. Both the Pfizer and the Moderna vaccines are 2-dose vaccines. Pfizer is given 2 doses 3 weeks apart, and Moderna 4 weeks apart. I didn't go through all of the efficacy data with both of those vaccines, but I think what's important to note is that you actually start to see efficacy even prior to the second dose, but you need both doses to get the high efficacy that the trials demonstrated. So a little bit different demographics for the Johnson & Johnson vaccine, because this trial was conducted in the US, South Africa, Brazil, Chile, Argentina, Colombia, Peru, and Mexico. And when you look at enrollment, only 44% of people were enrolled in the United States. So we have a much greater diversity of participants in this trial. Only 58.7% were white. More than 19% were African American or Black, 45% Hispanic or Latino, and greater than 40% with one or more comorbidities, which I think is important. So we have a much greater global population in the Johnson & Johnson vaccine trial. When we look at efficacy induced by the Johnson & Johnson vaccine to COVID-19 of any severity was lower than what we're seeing with the mRNA vaccines. But again, I think that goes to the global sort of population and the different strains of virus that were seen by participants in the J&J vaccine trial. So if we look at against disease of any severity, 28 days after vaccination, so early on, 72% against moderate to severe critical, but again, 85 or 86% against severe critical, and 100% against hospitalization and death. I think that's very, very important, regardless of where the trial was done. Overall, when you look at all of the cases that were accrued during this phase 3 trial, the J&J vaccine was 100% efficacious against hospitalization and death, which is our key public health endpoint. If we look here, we do have lower efficacy against the South African variant. You can see 64%, 28 days after vaccination, and somewhat lower efficacy in Brazil at 68%. Very good efficacy, though, against severe critical COVID. And I think that is key. 81.7%, and again, 100% efficacious against hospitalization and death. In South Africa, where I'll go through the numbers, but where I think more than 90% of the cases that were sequenced, the viruses that were sequenced from that trial, were indeed the South African variant. Also, very high efficacy against severe critical COVID in Brazil, where a large proportion of the viruses sequenced from this trial were the Brazilian variant. So the good news from this is that although we know that neutralizing antibodies induced by these vaccines work less well or neutralize less well the South African variant, we're still seeing very high level of protection. And I didn't mention this early, and I should have at the beginning. It's important to note that the spike protein that all of these vaccines, the sequence of the spike protein that all of these vaccines use is from the original Wuhan strain. So it's, they have not been engineered to include either the D614 variant or any of the other variants, the UK, the Brazilian, or the South African variant. And yet they are still highly, highly, highly protective against severe COVID, hospitalization, and death. So if we look at this summary of the efficacy of the Johnson & Johnson vaccine, it has a lot of advantages in that it is single dose. This is the only real world data that we have on how these vaccines perform against any of the variants. We do see a bit lower efficacy against moderate to severe COVID in each of the regions where this study was done. But again, 95% of the cases in South Africa were due to that variant. And yet the vaccine was highly protective against hospitalization, severe COVID, and death. 85% effective in preventing severe disease and 100% effective against hospitalization and death, including South African and Brazilian variants. So this is very good news from a global perspective. And because, as I said, because these vaccines all, for the most part, include the same sequence for the spike protein, even though we're seeing lower antibody titers against these variants, I think we're going to see really very good efficacy against severe disease, hospitalization, and death. A new vaccine that's coming down the pike is the Novavax vaccine. It is a nanoparticle constructed from the full-length spike protein, pre-fusion stabilized. Both the mRNA vaccines in Johnson & Johnson also use pre-fusion stabilized spike. They have some phase 3 results from the UK and South Africa that were released January 28th. They don't have as many cases in South Africa, but They showed very high efficacy in the UK with more than 50% of cases from the UK variant strain, which was excellent. Again, they had lower efficacy in South Africa against mild, moderate, or severe COVID. But again, 92% of those were the South African variant. They only had one case of severe COVID, so they don't have estimated efficacy against severe hospitalization or death. But again, I think the trend that we're seeing is that we have lower efficacy against COVID of any severity induced by these vaccines against at least the South African variant and perhaps the Brazilian variant. The vaccines, at least Pfizer vaccine, we have real world data that it is highly effective and efficacious against the UK variant. We know that the neutralizing antibodies induced by all of the current COVID vaccines are less potent against the South African variant. We know that they're highly potent against the UK variant. And although they are less potent against the South African variant and perhaps the Brazilian variant, they're still able to neutralize that variant. And we're seeing that in real world studies where we're seeing less severe disease. They aren't able to prevent infection. We're still seeing less severe disease with these variants, but we are able to prevent hospitalization and death, which is excellent news. I think it's expected that the vaccines, all of the vaccines, even the mRNA vaccines, which haven't been tested against the South African variant in the real world, will have lower efficacy against symptomatic COVID caused by the South African variant. But I do believe they will offer significant protection against severe and hospitalized disease. We will, of course, need more real world data to confirm this. All of the manufacturers recognize the risk to these variant viruses and recognize that there will likely be other variants that will arise spontaneously as long as the pandemic continues. And the manufacturers have all announced that they are making what I call second generation vaccines, where they're including within the vaccine different either mRNA sequences or adenovirus vectored sequences that include the South African variant along with the original strain, so that they'll have greater coverage of the South African variant. But I think the good news is, as we wait for those to come out, that we're still seeing excellent protection against hospitalization, severe disease, and death with the current vaccines that we have. And I would say that it is extremely important that we don't stop production and try to switch over to newer candidates, that we keep producing vaccines and we vaccinate as many people as we possibly can, as quickly as we can, to really interrupt the pandemic and hopefully at some point get back to a new normal. All of these vaccines don't necessarily prevent transmission. I do think that they reduce transmission significantly. We're seeing some evidence of that coming out of Israel, coming out of the UK. But again, we need more data to really look at this more closely. But I just like to stress, and I know I've said it over and over again, I'm was very, very pleased to see the J&J data that look at the effect of this vaccine against the South African variant, because I think it is encouraging that these vaccines all will have significant effects against all of the current circulating strains, including the variants. And that is all I have. Thank you.
Thank you so much to all of our speakers for those wonderful presentations. This is the moment when we would all be clapping and being very excited and jumping up to the microphones to send our questions in. So please continue to send your questions to the Q&A. But throughout the talks, we've been receiving some questions. And the first question that I have here comes from Rebecca Christopherson, and it's for Nathan. The question is, has anyone looked at the within host variability of sequences over time, both in general and with respect to variants of concern?
Thanks, Rebecca, for the good question. In terms of intra-host diversity with sort of non-variants, there have been several studies on this, and in general, we see very little within host diversity. I think this is represented also by the sort of slow evolutionary rate that we see of SARS-CoV-2, where we actually have transmission that happens faster than we have the accumulation of mutations. For the variants in particular, I haven't seen any data on this yet. I'm sure that there's some groups working on it. But I think that there could be, there certainly is a lot of genetic diversity that accumulates within individuals that have persistent infections or immunocompromise, where you actually, there looks to be like these like E484K evolves independently within a person. We can see those pop up. But once then we have transmission of these variants, their evolutionary clocks seem to sort of flatten back out, just like the rest of the viruses. So I wouldn't expect there to be increased diversity, but of course we need the data to look at that.
Thank you, Nathan. The next question is for Pei-Yong. The question is, do you have any information about the interaction of the D614 gene mutant with the innate immune response?
No, we don't. We haven't really studied that. But my speculation is probably not too much along those lines. I think it's mainly contributing to the conformation of the receptor binding domain to the open conformation, and that is more ready to engage the receptor and the infection.
Thank you for that. The next question is for Anna, and this comes from one of our anonymous participants. It's a two-part question. Is there any concern about the long-term safety for any of the new or now or soon to be available vaccines? And if you had all of the vaccines in your fridge or freezer, which one would you choose considering the benefit risk profile?
So I'll start with the second question and then go to the first. I would take any vaccine that were available to me right now. I will say that when I say that, I'm talking about the ones that we have efficacy data on. There are some, and this is my personal opinion, we haven't done a study here in the US to see, I'm less of a fan of the inactivated respiratory virus vaccines, mostly for the reason that I stated earlier, in that they don't really stimulate a very good CD8 cellular response. So I'm a larger fan of the adenovirus vectored and the mRNA vaccines. I would take whatever vaccine were available. We do look in terms of, are there special populations where one vaccine may be better than another vaccine? And one of the things that we're looking at with the J&J vaccine are people where it's really difficult to get them a second dose. So people who are in transient housing, people who are hard to reach, you may only have one contact with them. That is a population that I would prioritize for the J&J vaccine. The other interesting thing, and I have to actually go back and look at this, you know, none of these vaccines was studied in pregnant women. However, J&J does have an Ebola vaccine that uses the same platform as this vaccine, and there may be data on that vaccine in pregnant women that we could help inform us about safety of the vaccine candidate in pregnant women. So we are administering the mRNA vaccines to pregnant women. And so far we haven't had a safety signal. But again, I think that's a population that needs more study. And then in terms of long-term safety for these vaccines, so the mRNA vaccines, this is the first time they have been approved, formally approved, even though it's still under emergency use authorization. We will be following volunteers for 2 years to look at long-term safety. We certainly right now don't have any signals that would be concerning for long-term safety. Adenovirus, we've had a live adenovirus vaccine for many, many years that doesn't have long-term safety concerns. But again, even the adeno-vectored vaccines, we will be studying for 2 years as part of these trials. One of the early concerns with any COVID-19 vaccine was the risk for enhanced disease in subsequent seasons. And that's why all of these vaccines have to demonstrate a good Th1 response as opposed to a Th2 response to help reduce that. So people will be looking for that in subsequent seasons. We've not seen any indication of enhanced disease in any of the vaccine trials. And I think Mauricio wants to comment.
Yes, and I've been involved in J&J trial also, and also in the CoronaVac, the inactivate Chinese vaccine. In our center, we already vaccinated maybe 1,500 people in trials. And now over 6,000 people in my institution in regular use of coronavirus and the AstraZeneca also vaccine. So I'm quite familiar with using this. And I agree with you about the some issues regarding the inactivate vaccines, but it's what we have right now. We have a few million doses, and we are using with quite successful, I can say at this point. I was vaccinated with one. It's the only one that I have the chance to be vaccinated. But I like to add that I'm pretty sure that J&J are starting a trial soon with pregnant women.
Yes, yes, they are. They have that planned. And I just want to reiterate what Mauricio said. If you have any vaccine available, take that vaccine. Do not wait for a second generation vaccine. Do not wait. The most important thing we can do now is vaccinate people as quickly as possible. So please take whatever vaccine is available to you.
Thank you. The next question is for you, Mauricio. And is there any data on whether the new P1 variant is being shed longer in patients?
Not that we are aware. In Brazil, we are not checking these. Actually, we are checking these right now, but I don't have the data right now. I would like to say that we are under-reporting these, and we are not sequencing as much as we can because the companies are not delivering supplies as fast as we need. You all probably understand that there is a rush in the world to buy these reagents, and the companies are not being good enough to deliver it out here. So I'm sorry that I cannot answer this right now.
Thank you. There's another two-part question for Anna. Nathan, if you want to jump in as well, I just saw you turned on your video. Go ahead. Okay, so there were two confidence intervals that seemed to be very wide. The first one was for the Moderna efficacy against over 65. Is this the result of sampling factors or any other factors that could explain this? And also for the J&J vaccine efficacy confidence interval for severe disease appeared to be negative with PCR from any source. So was that true? And also, can any other factors explain this?
So this all has to do with the number of cases that you have that meet that definition. So when you're doing a trial, and again, they broke it, I think in that slide, that's broken down into different regions, which means you're doing subgroup analyses in an already smaller group, which is why you get those negative confidence intervals, is because you have a smaller number of cases in each group, and that widens your confidence interval. And that's the same for Moderna as well. It has to do with, they're looking at, for primary interim analyses. And again, remember what's been published and what the EUAs were based on was an interim analyses. It's not the end of the study. They're continuing to acquire cases. But at the time those data were published, you're looking at a total of about 150 cases. So, and that's symptomatic cases. That's not severe cases. So of that, even fewer are symptomatic. So for instance, in the Pfizer study, I think Pfizer only had 9 severe cases that they worked with. There were more severe cases in the Moderna study. But it is all a numbers game. And those numbers are going to change as more cases are accumulating throughout time.
Thank you, Anna. The next question is for Nathan. Again, it might be a two-part question here. What factors contribute to proliferation of newer strains of COVID generally, and in particular, the one dominating in South Africa? And 2, what kind of experiments and studies need to be done to further understand what makes variants more transmissible on the virology and pathology side of things?
Yeah, okay, so on the first one, this, just to reiterate, the question is, what is the evidence that we would have that they are more transmissible, right? Especially the South African variant?
Correct.
Okay. So I would say besides B117, we really don't have a lot of good data about other variants of concern or variants of interest, and actually if they are more transmissible. There's a lot of factors that can go into play here. some of it may be super spreader events that happen, and it happens to be with one variant, and we end up, through more of stochastic reasons, we end up sequencing more, detecting more of one variant than another, and it gets to higher frequency. There's clearly reasons for us to be concerned with B1351 and P1 in terms of vaccines, as Anna has explained, but there's and perhaps even reinfections, and hopefully that these vaccines actually are protective, as the data is suggesting, at least for severe disease. But in terms of transmission, it's still really uncertain. What we had for the B117 that we've looked at both now in the UK and the United States is that from multiple different locations, we have clear evidence of doubling between every 7 and 10 days that just through frequency itself, and also through the Public Health England doing the comparison of secondary attack rates. I mean, we need a lot of epidemiological data to really suggest that they're more transmissible. In terms of the lab experiments that need to go with them, they help to support, but really don't supplant the epidemiological data that we need. So, Pei-Yong did an excellent job of explaining a lot of these different assays. Competitive fitness assays are great of showing that one lineage is able to out-replicate and out-infect specific cells that another one cannot. That it's really just a combination of a lot of different data, both in vitro and vivo and epidemiological, that we need to make these assessments.
Thank you, Nathan. We have 5 minutes left, and so I'll leave you with one final question for all of the panelists, and I would like if we could start with Pei-Yong. What do you recommend that scientists should do to fight misinformation and alarm that is being disseminated by the press, particularly related to the emergence of mutants and efficacy of vaccines?
Well, I think the scientists, besides doing the science, also we need to disseminate the knowledge to the public and educate, the people around us. And also, I think it's equally important to influence the politicians so that their policies and their comments were in line with the facts. That's, I would say, we should do.
If there's nothing else on that one, then the other final question we could wrap up with is looking at the diversity of the demographics of the studies that have been conducted on the vaccines thus far. How can we ensure that there's equal equity or even greater equity in the rollout of the vaccines?
I can start with that because that's something we're all very much aware of and very much working on. One of the great concerns is that there will not be global equity in terms of getting these vaccines, and that we will have vaccine nationalism. And there are different groups that are working toward trying to ensure that there is enough vaccine, particularly for low income and some low and middle income countries as well. It's a huge problem. And, we need to produce more vaccine. We need to make more vaccines available. But until that happens, each country has to be willing to set aside vaccine for the global population. And right now, the US has sort of committed to getting vaccine for 450% of its population, because they were doing we don't want to put all of our eggs in one basket scenario. So they've committed to purchase enough doses of vaccine for 151% of the US population. The question is, and this is true for other countries as well, Canada, the UK, other countries. And what we have to do is somehow convince everybody within the US that, we're not going to get back to normal until everybody's back to normal. We're not going to travel until other countries have controlled this pandemic as well. And will we be willing to give up our extra vaccine before we vaccinated everyone? And I think, from the public health community, this is a message that we really have to get out. We're having trouble with equity within the US population, much less outside in the global population. And it's something that is going to require a lot of work. We've done a tremendous amount of community outreach, just not only for enrollment in vaccine trials, but also for vaccine distribution within the city of Baltimore. And it's something that has to continue. Every country has to be willing to give up some vaccine to somebody else so that everybody can get out of the pandemic. And I'll get off my soapbox now.
Thank you so much for that. Any of the remaining questions we can pass on to the speakers because we are at time. I just want to take this final opportunity to thank all of our speakers and participants. It's been wonderful to be together today, and we look forward to being together again soon next year.
Okay, thank you, everybody.
Thank you for organizing this, Patty.
Okay, thank you. Thank you. Fantastic.